While Griffith was able to show that there's something being passed from the dead S to the living R that transformed the R into a killer, there was great debate at the time as to what's being passed. You know, some camps said it's the chromosome, it's, DNA. Still don't know what DNA is, still don't know how it actually works. Others are saying it's RNA. Don't know exactly what RNA is, how it works, what its structure is. We just know it's there. Another group was saying, well, it has to be proteins. Okay, proteins are being passed, and it's in the proteins that compose the chromosomes that the information is stored. So there's these big arguments, and no one really knows what's going on. You have the work of Morgan, you have the work of Griffith. So we know the chromosomes have something to do with it. We don't know exact structure. We don't know exact function. Griffith showed that there is something that can be passed, and it carries what we now think of as genes, instructions, and how to do stuff. Well, fast forward next decade, the research group composed of Avery, McCarthy, and McLeod, they re-examined Griffith's work, but they deconstructed it. They went into this examining all the different arguments, and the arguments that it's maybe something lipid being passed from the membranes or the surface of the bacteria. Okay, or it could be proteins, it could be RNA, it could be this DNA stuff. Remember, we don't know exactly how DNA, RNA works, we don't know how the chromosomes function, but we just knew it had to be in one of those components. So, what the research group of Avery, McLeod, and McCarthy figured out was, let's remove each of those components stepwise, one at a time, and let's see what happens. If we remove proteins from the dead S solution, if proteins are what's transforming R into a killer S, if we remove the proteins, nothing should happen. If it's the DNA stuff, we remove the DNA, R should not be transformed. And this was the logic they had when they designed their experiment. They took a bunch of the S strain of the bacteria, and they heat killed it. Made a solution of boiled S strain. Okay, broke it all up. Same exact thing that Griffith did. And then they aliquoted that out into different tubes. Tube 1, 2, 3, 4, and 5. Tube 1, they added what is referred to as an enzyme that would destroy all the polysaccharides. Okay, remember polysaccharides are long chains of sugars. So it's an enzyme that's going to go through and chew up all polysaccharides. So they treated the S solution, and then they mixed the S with the R, just like Griffith did, and then they injected it into the mouse. Mouse dies. They took tube 2, mixed tube 2 with a lipase. Remember, rule of thumb, anything that ends in A's is an enzyme that acts on whatever prefaces A's. So a lipase is going to go through and chew up all the lipids. So Lipids destroyed. Mix the S solution, treated S, with the R. Inject it into the mouse. Mouse still dies. Okay. Tube 3. To destroy the RNA, you're going to use an RNase. Okay, it's an enzyme that goes through and chews up nucleotide by nucleotide any RNA present. Took the treated S solution, mixed it with R, injected it into a mouse. Mouse still dies. So, nothing yet. To go after the proteins, use a protease enzyme. Take the S solution, add the protease, allow it to digest all the proteins, mix that with the R, inject it into the mouse, mouse dies. So far, polysaccharides don't transform R. Lipids don't transform R. RNA doesn't transform R. And proteins don't. The one thing left, DNA. Go through, added DNAs. DNAs will chew up what we now think of as the double helix. So the double helix that was still in the dead S gets chewed up. The treated S solution mixed with R. That R is now injected into the mouse. Mouse doesn't get sick. Mouse doesn't die. Ding, 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 ding. Proof positive. DNA. Still don't know its structure. Still don't know exactly how it functions. But it's the DNA of the chromosome that is what carries the information.